I'm Vivian Li, an assistant professor at Rutgers University, Department of Biostatistics and Epidemiology. I would like to introduce our recently developed method named MAAPER, which is for model-based analysis of alternative polyadenylation using 3' end linked reads. This is joint work with the Bingtian's lab at the Wistar Institute. The polyadenylation refers to the addition of a poly A tail to an mRNA transcript, and APA is a widespread mechanism of gene regulation that generates distinct 3' ends in mRNA transcripts. And APA occurring in the 3' untranslated regions can lead to the production of mRNA isoforms with different stability and can also affect mRNA translation and localization. In human genome, APA occurs in over 70% of the protein coding genes, and it has been recognized as a critical regulator of gene expression. In addition, APA profiles are frequently altered in immunological and neurological diseases, as well as in cancers. Therefore, it is a critical task to analyze and summarize APA profiles from RNA-seq data. As many single-cell RNA sequencing technologies generate RNA reads from the 3' end of the RNA molecules, such data provide a nice opportunity for studying alternative polyadenylation. In particular, we focus on technologies that generate the so-called 3' end linked reads. In this scenario, the cDNAs are generated from the full-length mRNA transcripts and then fragmented. Next, only the fragments correspond to the 3' ends of the mRNA transcripts are retained for sequencing. However, in the sequencing step, the reads are generated from the 5' ends of the cDNA fragments. After being mapped to the reference genome, most 3' end linked reads have a distance to the poly A site. As their distances to the poly A site is random, the 3' end linked reads do not provide non-ambiguous information about poly A site positions, and we need to predict the poly A sites using a statistical method. Example technologies that can generate 3' end linked reads include 10x genomics, DropSeq, and also bulk sequencing methods such as QuantSeq forward and PetSeq. This is a diagram showing the major steps of our MAAPER method. It takes the mapped reads from two sample groups and the poly A site database as the input, and it has two stages of modeling. The first stage is to predict poly A site positions using a likelihood model, and the second stage is to perform APA analysis based on statistical testing to evaluate which genes have differential APA events between the sample groups. In the interest of time, I will show one application of MAAPER to single-cell RNA-seq data. We applied MAAPER to 10x genomics data from 5 placental samples and focused on 3 trophoblast cell types, which are specialized cells of the placenta that play an important role in embryo implantation and interaction with the maternal uterus. And because VCTs are the precursor cells of the other two subtypes, we compared the APA profiles of EVTs of SCTs with those of VCTs. MAAPER on average identified around 13,000 expressed genes in the 3 trophoblast cell populations, among which 44% had a single poly A site. And we find that around 75% poly A sites were in the 3' most exon, and 21% were in the introns. Using FDR-adjusted p-value equal to 0.01 as the cutoff, MAAPER detected significant poly A site changes in around 5,800 genes between EVTs and VCTs and 4,800 genes between SCTs and VCTs. The RED is a score we define based on poly A site quantification, and a positive score indicates RNA transcript lengthening, and a negative score indicates shortening. Interestingly, the genes with significant APA changes in EVTs or SCTs did not overlap substantially, indicating distinct APA regulation in two cell types. For example, MAAPER predicted 3 poly A site in the 3' UTR of MUC15. We find that this gene is not only differentially expressed between the 3 cell types, but also displayed 3' UTR shortening in the differentiation of SCTs. The MAAPER method has been implemented as an R package available at both CRAN and GitHub. If you have any questions about our method or software, please feel free to contact me for more information. Thank you very much.